Straight to it, lights. It looks okay.
Whichever way you want. So yeah, welcome everybody, and
Right.
thanks for coming to Drew Ellis's honors thesis presentation. And I met Drew. Gosh, when was it? You showed up in my office asking about the Native Plant Society and wanting to start a student club. So Drew has been the leader, fearless leader of that Native Gardening Society on campus. How many people are in that? There's few, I guess. I've seen some folks before. And at that point, Drew was working with Dr. Neufeld, trying to develop an honors project. And then he took a look at what we were doing in the lab and got really a lot more interested and excited about that.
Yes,
and switched. So we were lucky to have Drew working on a project he's going to talk to you about today. And he's done a lot of work, not just in the lab, but also bioinformatics, trying to figure out the processing for all the data that we got out of this. So yes, I'll turn it over to Drew. We've got a couple more seats over here if you want to sit. I can remove my stuff.
Thanks for coming, everybody. I really appreciate the support you're showing today.
Yeah. What wants that?
I'm gonna go ahead and turn down the lights just a bit. Just so it's as visible as possible. But I want to say thank you to Dr. Rose for those kind words. And also just before I begin, express my gratitude to Dr. Rose for her unending patience throughout my thesis. There were quite a few things that obviously I needed help with as an undergraduate researcher. And she was very amenable, kind, and patient in helping along with that process. And I'd also like to extend my gratitude to my second reader, Dr. Christian, for agreeing to give her time and participate and help me out with the process of writing as well. So thank you to you both. Without further ado, I'll go ahead and introduce my thesis. I'm going to read off the title. In this thesis, I am analyzing the fungal endophytes from the shining Firmoss in different seasons by isolation, identification, and next generation sequencing. So let's get into what that means. So to begin, I'd like to take a quick poll. If by a show of hands, who thinks that bacteria or fungi are bad? One brave soul. That would be false. In the most recent research that we've seen, there's been a big breakthrough in understanding the microbiome and its impacts on host microbe relationship health. So it's well documented, right, that the human gut contains a microbiome of bacteria that's it's extremely important to your overall health and well-being, and it assists with a lot of physiological processes. And the same is true in plants. So plants also host a wide array of bacteria, fungi, sometimes viruses and protists. They interact with I'll say thousands of different types of microbes in the environment on a day-to-day basis. And these host microbe associations are instrumental in determining overall plant health and success. So I want to first define what these microbes are characterized as that we're looking at specifically. So there's pathogens. Pathogens are known to cause disease. These can cause chlorosis and plant death with a whole wide array of symptoms. However, the ones that we're looking at are specifically characterized by their mutualistic or commensalistic style relationships. So these associations can either have no effect on the plant whatsoever, or it can also have an extremely beneficial positive effect on the plant's growth and survival. And these are called endophytes. Over here, I just broke down the word endophyte, endo meaning inside, phyte meaning plant. So it's living inside the plant tissues specifically. And these endophytes can be extremely diverse. These have co-evolved with these plants for millions upon millions of years. And through that coevolution, there has been diversification of different types of endophytes, and as well as diversification of the endophyte function within a plant. So endophyte function within plants can produce quantifiable, measurable effects on the plant's overall growth and success. That includes nutrient acquisition. It can assist in nutrient uptake, including nitrogen, phosphorus, potassium, all very important things for plants. They can produce bioactive compounds called secondary metabolites. These compounds have profound physiological effects in the plant. Also protection against biotic stresses. This includes pathogens, viruses, any other type of microbe that can pathogenize or cause disease in the plant. They can outcompete or sometimes even allelopathically kill and prevent infection. They also can regulate plant development, activate the immune response in plants. Few people know that there's actually an immune response in plants. They can produce siderophores, that allows them to acquire trace metals in the environment, which is extremely important for the plant's health. Also protection against abiotic stresses. So that includes low or high rainfall, includes high or low temperatures, includes salt content. So all of these factors can be attributed or assisted by endophyte function. Thank you, Dr. Christian. I was just thinking about that. So all these functions, this diverse array of functions, have been studied extensively, or not extensively, they've been studied for endophyte function in plants. However, a budding area of research has been focusing primarily on secondary metabolite synthesis. These secondary metabolites, like I said before, are bioactive compounds. They have physiological effects in plants, but also when administered clinically, they can have effects such as tumor reduction. They can help improve cognitive impairment in Alzheimer's patients with neurodegenerative disorders. So these endophytes are a source of novel bioactive compounds that can be used and applied at a broader extent for our society. That's why they're of interest to study. And so I want to highlight a plant. It's called the Huperzia serrata. This plant is native to China, and it's been used in traditional medicine to treat ailments such as contusions, swelling, things even so far as schizophrenia. And that's been used in traditional medicine for many, many years. So the demand for this plant has led to harvesting. And given the fact that this plant is extremely slow growing, just for a little bit of context, this leaf is probably the size of the tip of my thumb, indicating that this is likely 7 to 10 centimeters long, probably a 10-year-old plant, just about. Very slow growing, very small, not growing, it doesn't grow very fast. So harvesting the plant has led to concerns, has raised concerns about the sustainability of harvesting this plant for its medicinal properties. And so, which has also led to analysis of metabolites within the plant. And one of the important metabolites that's been found inside the plant is Huperzia A, which can be found in dietary supplements. You can order this online. So this metabolite does have important physiological properties and has been explored clinically. However, the issue is, like I said before, overharvesting has led to population damage. So we're looking at specifically concerns regarding sustainability of researching or using this plant medicinally. That led us to, in the past, look at Huperzia lucidula. Huperzia lucidula, oh, I want to mention this. This is an important concept. The plant itself contains fungal endophytes specifically that produce Huperzia A in large part or in total part for the plant. So these fungal endophytes, when isolated outside of the plant, can produce Huperzia A, which has led to research and regarding endophyte cultures and fermentation outside of the plant to hopefully have a sustainable alternative source of these secondary metabolites. So we looked at Huperzia lucidula. This is not native to China, it's native to the Appalachian region. It is the shining Firmoss. This has been explored in our lab in previous years for its similarities to Huperzia serrata, and it's been determined that the fungal endophytes, when cultured outside of the lab or outside of the plant in the lab, also produce this Huperzia A. So pretty cool finding. However, whenever they cultured the fungal endophytes outside of the plant, they did notice variable success rates. So it wasn't given whether or not they were going to see a high number or low number of isolates or types of fungi or numbers of fungi that they can grow and study. Which leads us into our objective for the study. So we were trying to determine what's affecting this isolation success rate, and we're measuring that in numbers of fungi that we're getting outside of the plant. with the idea that some factors might explain this variation, such as seasonality. So such as seasonality and also tissue type specific factors. With the implication of, if we can determine what's causing this variable success rate, maybe we can target these specific seasons or tissue types and be able to more sustainably research these endophytes producing metabolites with a high success rate and reliable data at the same time. And so I just wanted to showcase some or define these specific regions. So tissue types, we're using the tip. So this is one single plantlet. And I'm going to show this again, but I want to just clarify some things. This is one single plantlet. We separate on the tip, stem, and rhizome. So remember those words. I'm going to use those quite often throughout the presentation. And then also seasonality, we're sampling based on seasonality. We'll see that in a second as well. Oh gosh. Okay, I copied and pasted that figure, so that was an accident. Okay, so to do this, we actually need to sample, we need to culture these endophytes outside the plant. So to do so, we did seasonal sampling in the summer, fall, and winter. I wanted to do so in the spring, but time limitations applied. And again, we're seeing this figure is sectioning based on tip, the stem, and the rhizome. Those are 3 different sections throughout the plant. Then after that, we surface sterilize the plant itself. The plant has surface living microorganisms that can contaminate the sample, so we want to make sure that we're getting rid of those and killing those off. I'm showing bleach, but it actually was a sequential sterilization procedure of ethanol, bleach, tween, hydrogen peroxide, but I'm just saving room. And then after we got this plant sterilized, we then sectioned them into about 1 millimeter increments. We placed them on potato dextrose agar plates, which is a widely accepted way of culturing these fungi. And once we saw the hyphal emergences of those little fruity hyphal mycelial bodies, I then was able to plate them on a separate plate to get them by themselves or in an actinic culture. And once I achieved an actinic culture, I took the DNA from those fungi. I performed amplification of the ITS 1 to 2 region using polymerase chain reaction. And if anyone's not familiar with ITS 1 to 2 region, it is a section of DNA that's highly useful for assigning species to the plant itself, just to identify what you're actually looking at. Once we had those PCR products, we sent them off for sequencing and the forward reverse reads. And the forward reverse reads were able to give us a consensus sequence where we compare them together and get the most reliable data we can. And we aligned them using NCBI BLAST Plus for taxonomical identification. And then finally, tying it back to the original points of what we're looking at, we tested for seasonal and sample type differences using a quasi-Poisson generalized linear modeling. And this is just a quick schematic of the ITS 1 to 2 region. If anyone's not familiar with things like this is DNA right there. And this is ribosomal DNA, which is found throughout a large portion of the genome. And if you zoom in, you see a section of the rDNA. And if you zoom in again, right here, this is what we're sequencing. This is specific to fungi. It's useful for species identification. It's about 500, 600 base pairs long. And so that's what we're using to identify these fungi. This is a big table of a lot of information that you don't really need to know. So I'll showcase some specific information from it. This is representing our 67 isolates that we saw across 3 different seasons for all the different types of tissues with assigned species from NCBI. And with the assigned species, this is the alignment that we had from the ITS 1 to 2 region that was sequenced. And so what that means is just what's the overlap that we're seeing in these sequences and the type specimens. And if anyone's not familiar with it, typically there's a standardized level of which you want the sequence to be to assign an actual species, and we're not seeing it at that level, just indicating that we're isolating something specific that hasn't been described before. And this is likely not actually the species. But for our purposes, we're counting anything within plus or minus 1% as a single species, and that won't affect our statistical analyses downstream. All right, so this is a fun bar plot, and I'll just do a quick overview of what this is showing you. So it's separating our samples based on seasons at the bottom, fall, summer, and winter. And if you recall those sections of tissue types, it's RST for rhizome, stem, and tip, and that's for each season, okay? And it's colored based on rhizome, stem, and tip as well. And these bar plots are showing you the results of the generalized linear model that we use to determine statistical differences across our samples. So using the generalized linear model, we were able to create an expected mean number of observations per isolate. So what that means is that it took what we saw in each season and sample type and gave us results based on the linear regression that is able to create based on what we saw. So this is showing you the expected number of isolates, and I'm just going to showcase some of these trends that we're seeing. In the winter, we see an increased number of expected mean number of isolates compared to fall and summer. So you can see this visual representation of an increase based on the season, and this is in fact significant. So what that means is that we're seeing a higher number of isolates, and it's expected that if we perform more samples, you will see an expected number of isolates at a higher rate compared to the other seasons. So that's a significant finding. And then another trend that we're seeing is if you look across the sample types within a single season, these are larger compared, the tip is has a higher expected mean compared to rhizome and stem, which has a lower expected mean. And this is true across all 3 seasons, right? And so that's also a significant finding. So what does this tell us? It's telling us that perhaps that we're seeing an increased success in the winter, and that's independent from the sample type. So it's likely recommended that for future studies that are prioritizing fungal isolation success, the number of fungi that we're seeing at any given sample, that we should prioritize sampling in the winter, and if not in the winter, then at least from the tip, right? Because that's going to give you more higher volume of fungi to look at and characterize and study. Moving on to another fun graph or figure. This is showing you all of the species that we saw and plotted against our sample types. So this is again separated by season with the fall, summer, and winter, as well as by sample type, rhizome, stem, and tip for each season. Okay. And so just to highlight that statistical analyses can be limiting sometimes to what you're actually seeing. So let's look at winter and tip for the first. We're seeing obviously this increased. Oh, I should also specify the red color is associated. Red, bigger number. Bigger bubble means bigger number. Blue, smaller bubble, lower number. Okay, so we do see this increased number based on the size of the bubble and the color. In winter, I think it's about 20 observations, right? But compared to the winter and the stem, we're seeing only 2 species in the winter and tip, as opposed to 7 different species in the winter and stem. So we're seeing a lower diversity in the tip, even though we're seeing an increased amount of fungal isolates we're actually able to culture. We're not seeing a greater diversity, and this is true. And this is just the overlap of species. So whatever is seen in the tip is also seen in the stem. It's represented. This is true in the summer as well, where we see 3 different types of species at variable rates, as opposed to stem, which sees 4. And this is not true. I want to showcase in the fall. In the stem, we see 1 as opposed to 3 in the tip. So not necessarily confirmed that there's a correlation between diversity and sample type, but there is a general trend that maybe we should explore in the future. And therefore, based on this trend that we're kind of seeing, perhaps studies that are prioritizing fungal isolation rates, the number of fungi that we're actually able to have in lab and study, should prioritize sampling in the winter in the tip first. But if you're looking at comparative analysis, looking at different types of fungi, maybe like different characteristics of the fungi, sampling the stem as well is also possibly recommended. And maybe hopefully leaving the rhizome alone. There is some representation of species in the rhizome that you don't see in either the stem or the tip. However, there is a great amount of variety and diversity in the stem and tip, and hopefully you can leave those root systems alone and allow it to continue to grow after you actually sample instead of killing the whole plant. So that's what we found for our culture isolation success. However, when you culture in lab, this is kind of limiting like I said, there are 67 isolates representing 11 different species. And within any one plant at a given time, you can see hundreds to thousands, I don't want to say thousands, you can see a lot of a great diversity of species in a plant at any given time. So how do you bypass this limitation, this limiting perspective? Well, we used a high throughput technology such as next generation sequencing. And what this allows you to do is compare DNA across the entire sample, and you can identify it on that ITS region that I showed you earlier. You can identify as close down to species as you can, and do a comparative analysis based on what's actually in the community at large, as opposed to what is present that you can culture. All right, so to do that, we sampled in summer, fall, and winter, same time, same days, same conditions. Sectioned it the same way, and it was from the same plantlet. I don't know if you noticed, but the tip is switched, and the stem is switched, and the rhizome is switched. So we used whatever wasn't left from the culturing experiment for our next generation sequencing. And then we also performed surface sterilization at the same time in the same way. Often I had to store it and freeze it until I could actually extract genomic DNA. I quantified the DNA content and estimated purity just to make sure it's within acceptable ranges for sequencing. Sent it off to someone else to do it for us. So they did ITS1 amplicon sequencing using Novasec 11 platforms. Then we input our raw FASTQ files using containing all of our sequences into our RStudio pipeline for analysis, and then tested for difference in season and sample type, similar to what we did for our cultured isolates, using RStudio as well, using a permanent lab. Okay, so what do we see? Okay, well, if anyone's not familiar with Shannon and Simpson diversity indices, I definitely didn't when I first got into it, so I'll explain as best I can. These are measuring two different things, and they both measure them in different ways. So measuring richness. So richness is the amount of fungi that you see. So if you have 10 versus 100, that 10 number is going to drive those numbers lower, and that 100 number is going to drive those numbers higher. Okay, so it's based on richness, the diversity of different types of species that you're seeing. And evenness is showing you the dominance of taxa. So if you have those 10 species and they're each represented 5 times in your sample, that's going to be high evenness. It's going to drive those numbers higher. And if you have those 10 samples and one is represented 5 times, whereas each other one is represented only once, that's going to be a high dominance, which is very common in microbial communities. Microbes can often outcompete each other for resources, thereby driving other populations down because they're consuming all the resources in the environment. So what do we see? We saw high Shannon. Oh, I want to also say that this is prioritizing richness. This is prioritizing evenness, even though they both kind of measure the same things. So the highest Shannon's diversities, the highest richness that we're observing is in the winter. And the highest evenness that we're seeing is in the Simpson diversity for winter stem and tip. Okay, that's pretty interesting. If you think back to our cultured isolate success, we saw an increased number in the winter. So it's not confirmed, of course, but there may be something to do with the richness and evenness of taxa that's contributing overall to the success rate of fungi that you're having in the lab. And then when we looked at the Shannon and Simpson diversity indices for fall, we did see the lowest numbers, which coincides with the lowest Simpson diversity, which is also interesting. I didn't show you the exact numbers for fall isolates, but fall compared to winter, we had 9 in the fall and 44 in the winter. So there seems to be some relationship between the number of isolates and the richness and evenness. I do want to highlight the fact that this is not confirmed. And the summer rhizome, we also see comparative numbers to the winter tip at 3.29 and 0.9, which is very similar, but we didn't see the same sorts of isolation success. So not confirmed, but it is an interesting thing to think about. And this is showcasing this diversity and representation. So if you look at fall across the bar plot, you're seeing a lot of dark and, oh, let me explain. This is a heat map, and whatever's red is assigned to a specific assigned sequence variant, and that's typically representative of a species that you're seeing. We had 1900s across the board, and this is representing the top 200 that were represented at the highest amounts. Okay, and where you're seeing red is represented at higher abundances, And where you're seeing this black blue, it's represented at lower abundances. So if you look at fall specifically, you're seeing more blue than red. Of course, you're seeing some red. That's given. It's going to have some species in there, obviously. But I wanted you to look at that and compare that specifically to the winter stem, which you see a lot more red across the board, across the column. I think you can maybe perceive that. Just kind of highlighting the fact that these species are represented at similar levels across the sample comparative to the fall, just for a visual representation of what I'm talking about. All right, so now we're moving on to analysis of our permutational analysis of variance based on season and sample type, okay? So this is a figure, and without getting into the nitty-gritty of the statistical analyses behind it, it is measuring difference, okay? So difference is based on distance. So the more distant something is from another object, the more different it's going to be, okay? So let's take an example. We look here, not as much distance between those two compared to that blue dot over there. Same goes for these two. This is, by the way, these are stem and tip samples. These stem and tip samples are more similar to each other and farther away from the rhizome, indicating that they're similar to each other, but distinct from their rhizome counterpart. Okay, and now we're looking along the PC1 axis. And I'm trying to explain it in increments because it can get really complicated really fast. The PC1 axis explains most of the variation. So whenever you take this Bray-Curtis similarity matrix, it identifies a principal coordinate that will allow you to plot differences. And this explains most of the difference. So what we see along the PC1 axis as we move left to right is that the seasons seem to be grouped together sort of and plotted separately within the PC1 axis. We can see that for the summer, the winter in the green, and the fall in the red. However, so it seems to be grouping these samples on seasonality, indicating that they're more similar to each other within the same season compared to other seasons. However, we don't see the same type of grouping with the sample type. For instance, this triangle, if you can see the triangle, is representative of a stem sample, and it's different from this stem sample, which is all the way across the board different away from this stem sample. And if they were more similar to each other, if the variation was being explained by sample type, they'd be clustered together, indicating that they're more similar. So we don't see the same type of grouping for sample type as we see for season. So let's move on to another fun table. Is this significant? So let's look at first season. Right, and so season is explaining 40% of the variation that we're seeing in the community at large within the species composition. That is significant. So we still have a significant value for difference in season. So anyone who's not familiar with statistical analyses, significance just indicates that this is a reliable finding that can likely be replicated across different experiments. However, for sample type, when we look at that, this is representing only 20% of the variation, which seems like a lot, but this is actually not a significant finding. So what is this telling us? Well, this is telling us obviously that there are factors affecting the community that we see across different seasons, but there's not differences between these tissue types. With season, this does overlap with our previous statistical analysis, just indicating that we're seeing differences in species composition. So maybe this difference in species composition can account for the number of fungi that we're able to culture in the lab, and selects for specific fungi that are culturable. So perhaps the winter season affects the types of fungi that are present in the sample by increasing the light, because in the winter you have no upper canopy and it's constantly performing photosynthesis, which allows for photosynthase being provided to your fungi that they can feed on and grow and reproduce and survive. Maybe there's a difference in the adaptability of these fungi. These fungi have to be able to adapt to different types of conditions. So adapting to a laboratory is no different than adapting to a different season. Also, it's surrounded by leaves, which bring endophytes down that live in the trees. And it's in the winter, it's surrounded by these leaves, and these endophytes can transfer from leaves into the plant itself. So maybe those leaves are more culturable. right? So there's a lot of different factors that we don't know that are explaining this variation, but one thing is for certain that this does overlap with our previous statistical analyses. However, with sample type, we don't see this difference, which is expected, right? Not everything's going to explain another variable. So in sample type, the factors that are driving the differences or not for our culturable success in the lab may not overlap with the difference in species composition that are driven across the entire plant itself. So maybe some things that are affecting increased isolation rate in the tip, maybe that the tip is exposed to the environment and the environment has fungi that are traveling everywhere, colonizing everything that they can. These are called generalists. And these generalists may be able to colonize the plant, but also pathogenize an insect or degrade a dead leaf tissue, right? So this offers them diverse functionality. It's also, again, more exposed to the environment, making them have, necessitate further adaptability. So lots of different things that can possibly explain it. Of course, that's not the focus of this research, but it is some cool things to think about maybe for the future. All right, so that kind of concludes what I have for my findings and everything that I determined in my sample. And so just to conclude, overall, future research should prioritize sampling in the winter and the tip for increased number of fungi that you can actually grow in the lab, and maybe from the stem as well, to get overall higher diversity of fungi for comparative analysis possibly. And future directions for where this project can go takes a wide standpoint, right? So we can do metabolite, microbiome, transcriptome analysis, and we can do that of the wild plant samples. And by doing so, you can determine these biosynthetic pathways, which can be extremely valuable to producing these metabolites on a wider scale, and possibly even correlate specific fungi that are growing in the plant with levels of Huperzia A or different types of metabolites that are of clinical interest. And the same thing can be done for plants that are grown in tissue culture. So looking at these levels of regulation across the DNA, across the proteins, across the RNA, can be also extremely valuable for understanding how this happens in the plants alone without the fungi. And also, if once you get those correlated fungi, perhaps we can isolate those fungi in the lab and introduce them to these sterile tissue cultures and see maybe what the difference is. That'd be kind of cool to look at, right? And then also, if you can regulate this environment and the tissue culture, perhaps growing a bunch of, if anyone's not familiar with tissue culture, you take like the, I don't exactly know the word for it, but these special cells from the plant, and you put them into a growth media, and it can grow just exponentially comparative to a plant that grows in the wild that has to go from a spore to a gametophyte to 15 years later, you have a full plant that's able to be harvested. So growing it all at once can maybe increase your metabolite success. So doing that analysis would be really helpful, maybe with tissue culture. And then also looking at the fungi by themselves can also be really valuable. That's why we culture them in lab. So we can do genomic analysis of biosynthetic gene clusters, and no one knows what that means. But it is a really interesting area of study possibly, along with the same types of analyses like transcriptome and metabolite analysis to determine the levels of regulation of these metabolites. And with that, I am just going to say thank you again, first of all, to Dr. Rose. Thank you so much. If you guys aren't familiar with my journey, I messed up a DNA extraction probably 3 times, which probably is like $300, $500. It's a $300, $500 mistake. So I had to source those funds somewhere. So Dr. Rose definitely was helpful in that process and just was super patient and understanding and a guiding force in my thesis.
Made him write a lot of practicals.
Yeah, yeah. So, and also thank you to everyone in my lab. These folks helped me with my sample collection. They've helped me label tubes and weigh tubes and just super helpful. I couldn't be more appreciative of them and their support. So Erica is here with us today. Thank you, Erica. And Alexa and Noah and Evan can't be with us today, but also Ethan is here. So thank you guys so much. And I'd also like to acknowledge the Office of Student Research, the College of Arts and Sciences, the Honors College, and the Biology Department at Appalachian State for helping fund this project. It unfortunately is a very expensive process. Thank you very much. Are there any questions?
I was thinking about your graph with the various sized circles that represented diversity and richness. Do you think in future research on fungal endophytes, like, what do you think would be more useful for research? Would it be diversity or volume, or does it kind of just depend?
No, yeah, absolutely. It really is dependent on the research that you're doing. So I think if you're trying to keep the same fungus in an alter its environment to really see kind of what is affecting these as a control in your experiment, right? You have the same fungus that you're working with, so it gives you a greater volume of fungi to work with, and you can expose it to a greater array of environments. Whereas if maybe you're comparing species, right, and these is essential for determining maybe like the highest level metabolite production. You can do comparisons between the species, so it's really dependent on what you're looking at specifically. Thank you. Hi.
So I am using like terms that I haven't used since high school, so excuse how I asked this question.
Yeah, of course.
I'm assuming you probably developed a hypothesis before going into this, maybe?
Yeah.
Cool.
Yeah.
I'm curious kind of what that hypothesis was and what surprises you had along the way, because this is a very extended project. You've been working so hard on it. What were you surprised by in the process?
Yeah, I was so going into the project, right, we're looking at differences in seasonality. We weren't originally looking at differences in tissue types in terms of fungal isolation success, so the number of fungi that we're getting. So what really surprised me was seeing an increased number of fungi in the winter, because if anyone's familiar with foraging, right, typically you go out foraging in the fall, and so we've kind of had a thought that, you know, increased moisture, lower temperatures, which are conducive to fungal growth, might increase that isolation success in the fall, but we saw it in the winter when things are dead, dying, and and decaying, right? So that was a surprise, but also we weren't looking specifically for differences in tissue type, but it did appear to be significant in terms of isolation success. That was really interesting as well.
Cool. Would you expect this to be, your findings to be applicable in a region that's not like specifically Appalachia or Western North Carolina? Or is it just very hyper regional because of how you did the testing?
Yeah, so I mean, with any scientific findings, you should really be careful with how you're generalizing your results. So I will say now that this can really only be applied to Huperzia lucidula and the high country, right? Of course, maybe some patterns of can be applied and studied and determined before really saying yes or no to that.
Awesome.
How do you feel like people in your field conducting similar studies are able to communicate results like this to the companies that make those supplements on how to be more sustainable? What's that interdisciplinary communication like? Have you seen any of that?
Yeah, that is an important subject that is, you know, widely discoursed in the scientific community. We do a lot of really complex analyses that are not accessible. So anyone who's really concerned with sustainability, right, I show this presentation to them and I can, you know, explain my permutational analysis of variance and my Bray-Curtis dissimilarity tests, and that means nothing to them. So at the end of the day, I think scientists really have to prioritize explaining and condensing the information in an understandable format, right? But I personally have not seen this discourse and interdisciplinarily, which I think it occurs. I haven't seen it myself, but it is very important going forward. Any more questions? All right, well, if there's no more questions, thank you so much, everybody. Thank you for showing your support today. It really meant a lot to me that everyone walked in the rain to see me talk to you. I know, I'm so sorry. It's like I try to turn down the thermostat.